Hi, I am Mrs. Sloan, and this is a video for my honors biology students, and we're going to talk a little bit about evolution. And this is a continuing topic for us. I want to remind you before we get started, get myself here in presentation mode, is that I have the notes down in the descriptor of the video so you can follow along, and column 1 is all the scaffolding for the notes, and then column 2 you can throw in little pictures and images that would be helpful. So we have already introduced evolution when we talked about early Earth, right? And we talked about all the way up to how cells could have formed. And so I want to review that really quick before we get started. You're going to need to know this when you take your final exam this semester anyway, so this will be a good little review for you. So remember when we talked about the steps of life and the ability for inorganic molecules to form organic molecules and macromolecules and polymers to form cells. So we went through all the steps of how that could occur and the energy it would take to make that occur, right? We talked about UV radiation from the sun. We talked about molten magma. We talked about radiation from the Earth's core, right? All of that would provide energy. We talked about Miller, right? And how he did what we had 1 billion years to do. He did in just a little over a week's time, where he took inorganic molecules, gases that you would find out of a volcano, right? Inorganic, and formed organic molecules. And then from that, you would need to hook these monomers together to build polymers, to build cells. Okay, so that's the first part of chapter 27. But in this unit, we're starting in on the evidence for evolution. What evidence do we have for that? All right, so the first thing I want to differentiate between is microevolution and macroevolution when you hear those terms. So microevolution would be a genetic change, so some change in a population. Like for instance, if you look at the human population, we've gotten taller over the years. Macroevolution would be a process where you would classify an organism as a different species. And we're going to talk a little bit later about how difficult that is to draw the line between species. So what I want you to be thinking about is, if you're going to have these change over time, where something is going to get selected for as being better than something else, then we need to think about where could this variation arise from and how could you pass that variation on. And when you're in my class, I will show you this video. All right, so the pieces of evidence we're going to look for that this change over time has accrued, and intermittently you actually would call them different species, come from these 5 pictures. So see if you can figure those out. I bet you can when you're looking at them. Here, I will just jump to it. We're going to look at fossils. So that was typically like that was the early thing, was to look at fossils. And now modern, we do a lot of biochemical evidence, comparing DNA, comparing proteins. We're also going to look at anatomical features. We're going to look at the geologic timescale and biogeographical evidence as well. So on your notes on what is evolution, evolution is, on your notes, genetic changes in a species over time. Genetic changes in a species over time, resulting in the development of genetic and phenotypic differences that are the basis of natural selection, because nature is selecting, right? And then you have descent of organisms from a common ancestor, And now we're starting in on the fossil evidence. That'll be the first one. Okay, so in your notes, fossil. A fossil is defined as the remains and traces of a past life, or any other direct evidence of a past life. It could be skeletons, it could be shells, it could be seeds, it could be insects, it could be imprinted. But in order to be classified as a fossil, you have to be at least 10,000 years old to be a fossil. And I've listed those things here. And fossils support the common descent, that you had a common ancestor and all these organisms have descended from that common ancestor known as Luca. So must be at least 10,000 years old. Now let's go through a few types of fossils. So you've heard of amber, probably with Jurassic Park and what they did with that, right? But amber is like sap from trees. So you might find insects in amber, but you're not going to find a woolly mammoth in amber because it's not going to get stuck to a tree. You could find a woolly mammoth though in ice. That might be where it would be preserved. You can look at petrified wood. There are petrified forests. So what you're looking at there is that the organic material of that tree have been replaced with minerals, but they still have their same outline, all right? And then here is a petrified salmon. Look, he has some nasty looking teeth right there. And then molds and casts. So this is another kind of fossil. So a mold would be, think of like a jello mold, right? Okay, so a mold, what would happen is you have this organism and you have dirt and mud that has formed around that organism. And then when that organism decays, you still have the outline. That is the mold. If some other dirt or mineral fills in that same shape, that would be considered a cast. All right, so there are molds and there are casts. And on your notes, you have types of fossils: amber, ice, petrified, mold / casts. And then here's an imprint. So this is the outline of a fish, right? Now think about it, it's pretty incredible to be a fossil, right? If you're going to get the actual creature itself, right? It has to be something that can be fossilized, but oftentimes their mark within the sediment can leave an imprint. Like if there have been fossils of footprints of dinosaurs or humans, then that would be considered a fossil as well. Oftentimes you find these fossils in sedimentary rocks. Sedimentary rocks are layers of sediment, probably coming from a body of water, and things get trapped within those sediments. And the assumption is the layers on top are newer and the layers on the bottom are older. Of course, you need to look to see if it's at a subduction zone, right? Where one bit of earth is going underneath another, right? So I think we have that on the types of fossils. So here is an example of a bulldog fish. Look, it's huge, and it was found in sedimentary rock. All right, so characteristics. What do you see when you look at the fossil record? And when you look at these time periods, we're not going to learn all of these, but we are going to learn these eras right here. Do you see on the side where it says Paleozoic, Mesozoic in the middle, and Cenozoic? You are going to learn those, and I'll talk to you about those in just a little bit. But look at when you look at the organisms, how they change over time. The oldest rocks you can find have the simpler organisms, right? And then here, look, you're looking at vascularization of plants where they can survive up on land. You're looking at fish with jaws and early reptiles, right? And then your first mammals. It's changing over time. And the earlier organisms are simple. And then those fossils that you find more recent, right, are a little bit larger, right, with more adaptations, multicellular, etc. And you can see a gradual transition in form over time. So characteristics of the fossil record: earlier organisms are more simple, and there's a gradual transition in form. And I'm going to give you an example of the horse, okay? The earliest fossils of the horse are small, almost dog-like. And the environment that they lived in was like a forest. So they were like running around in a forest. It was better to be small so they could jump under things or whatever and hide. But remember how the plates have shifted over time, right? You've got those convection currents shifting the plates around. And as a result of that, where they are on the surface of our Earth could change the temperature, the amount of rainfall, right, that they have. So horses, the areas they were in, gradually started being in areas where there wasn't as much rainfall, so you didn't have forests, you had grasslands. So it was better to be taller. So you see those fossils keep looking to my left. I don't know. I'm going to come right over here. You keep seeing those fossils then end up getting bigger and bigger because those are the ones that survived. Those are the ones that survived and could make more offspring. And they became the horses that we know them today as equus. All right, so that's your horse example on your notes. Now, as far as transitional fossils go, we can talk about that a lot, but for the purposes of our discussion, there are transitional fossils between fish and amphibians and reptiles. And here's a transitional fossil between a reptile and a bird called Archaeopteryx. So it has a lot of its reptilian features as well as those bird-like features. So transitional fossils resemble two groups that are currently classified separately. Now, should birds be different than reptiles? Probably not. But people are like, see these flying creatures and they're like, oh, that needs its own group. But anyway, we'll talk about that later. So resemble two groups that are currently classified separately. Oftentimes, these transitional fossils represent the intermediate. And the assumption is you were either better as a reptile or as a full bird, so you don't have a lot of those transitional fossils because they weren't successful. All right. And then I said, think Archaeopteryx. Now, dating. How does dating go? Well, I hope you can find dates. There's two kinds of dating, relative dating and absolute dating. Relative dating, I guess if you can't find somebody else, bring your cousin to the prom. That's not really what that means. But relative dating is you're saying this is younger and this is older. Why? You're in a layer of sedimentary rock and you're assuming the layers on the bottom are older and the layers on the top are younger. Okay, so that's relative dating. Then what you do is absolute dating is where you get the actual age of an event. So you're using like carbon dating or something along those lines. Now, keep in mind, index fossils, like there's only a certain time period when trilobites, this would be a trilobite, were around. And so you can use that also to date the layers above as being newer and the layers below being older. All right. So on your dating, relative dating is the order of events using sedimentary rock layers, And then absolute dating is when you're looking at the actual age of the event. So for instance, if you could take a radioisotopic sample from section A over here, and you could actually find out how old it is, then you could apply that to other samples that have the same sort of species within them. So how does that absolute dating go? Well, it's half-lives. It's how long it takes whatever you're sampling there for it to decay. And so you have a rate of decay that is known for different isotopes. So when you look at a sample and you predict, well, I think, today this percentage is this particular isotope, and then you find that you have a very, very small percentage of that isotope and you know how fast it decays down into that, then you can say it is approximately this many years old. So for instance, the half-life for carbon is 5,700 years. So if you had, for carbon-14, not carbon-12. So if you had a sample, right, and you could tell by looking at what percentage of carbon-14 is in that sample, you could tell how old that sample was. Did it go through 3 half-lives, 4 half-lives, etc.? So depending on what how old a rock is, you would have to use a different isotope. So on absolute dating, it is the actual age of the event, usually with radioactive dating. The rate of decay is fixed. We know how fast something ages, right? Okay, next. When we look at the geologic time scale, let me just put myself right here. Okay, when you look at the geologic time scale, this is a very simple picture, okay? I want you to look at the eras. I want us to focus on the eras right here, okay? We have Precambrian, okay, Paleozoic, Mesozoic, and Cenozoic. So first of all, to remember those, I think of Cenozoic as the center of your world now. Mesozoic's in the middle, Paleozoic is old and pale, pale old. And then everything before that is just called Precambrian time. Now, Precambrian time is huge. It is from the beginning of the Earth till about 542 million years ago, okay? So we're going from billions to millions. So this is a very long period of time. So look over here to the right, and I'm going to give you a way to always remember your eras and the years they encompass, okay? So beginning of the Earth is 4.6 billion years ago. You know the first fossil was about 3.5 billion years ago or so. So you can see that here on this chart too. Well, let me get a pointer. Oldest prokaryotic fossil is about, if it says 3,500 million, that means 3.5 billion, okay? So that's your oldest known fossil. Now, look what's going on. Oxygen revolution, eukaryotes, simple multicellular organisms. All of that took place in the Precambrian era. And you know when it ended? About 542 million years ago. Then your Paleozoic era, you have 542 million years ago is when it started here at the beginning of the blue. The end of the blue is about 248 million years ago, okay? And in the Paleozoic era, you have a lot of evolution occurring. You're going from algae all the way to giant trees. You're going from small multicellular organisms all the way to reptiles. So there's huge jumps that occur in the Paleozoic era. Mesozoic in the middle. Probably one of the periods that you know from the Mesozoic era is the Jurassic period, right? From the movie Jurassic Park, and that's when dinosaurs were flourishing. Now, they first started evolving reptiles in the Paleozoic, but they really gained their ascendancy in the Mesozoic era. And you can see, look at your time period here, 248 million years ago to 65 million years ago. Now, the era you're in right now, okay, that era is the Cenozoic, and it started about 65 million years ago, and it ended now. Oh, now, okay, now, right? It's still going, is that. So here's how I remember it. The era you're in right now, okay, a lot of you right now are learning how to drive a car. What's the speed limit on the freeway? 65, okay? It's the center of your world now. You're driving cars. The Cenozoic era goes back 65 million years ago. Mesozoic era ended 65 million years ago, but when did it start? 248 million years ago. You know what time that is? About the time school's out. Okay, then the Paleozoic era, right? It's ending 248 million years ago, but it started 542 million years ago, old and pale. It's about starting at dinner time. Okay, then you just need to go all the way back to the beginnings of the Earth, right? So all you have to remember is the Cenozoic era goes back 65 million years ago, right? That's when it started, and you think about driving cars. Then about the next era back, okay, Mesozoic in the middle, that's going back to about the time school's out, 248 million years ago. Paleozoic, about dinner time, 542, like 5:42 at night. You got me, right? Okay, and then you just have to remember the start of the Earth. Those are the only numbers you need to remember. Okay, so on your notes, you already have all the numbers up there. Now we just need to go over the basic species, and I'm going to divide them out for you. I want you to see, I'm just making note of this, that there are 5 major extinctions where sometimes up to 90% of life was extinct and had to start all over again. And so these extinctions, okay, would give opportunities for new species to adapt into those areas. So for perspective on the whole geologic timescale, let's go from 12 o'clock midnight last night, whatever day this is for you. I don't know what time of day that you are watching this video, but let's think back last night when you were sleeping at midnight. Hopefully you were sleeping at midnight. That will be the beginnings of the Earth, the formation of the Earth. So last night, about 2, 3 in the morning, 4 in the morning, 5 in the morning, that's when you got your first prokaryote, when you got your first cell. Okay, so midnight to 5 a.m. Okay, and we're on a 24-hour clock. So we're going midnight to last night to midnight tonight. That is when you got your first cell. Then about 6 a.m. is when you got your first autotroph, your first photosynthetic cell. It's 7, it's 8, 9, 10, 11, 12. It's all the way noon today. Okay, 1 p.m. all of a sudden you have the oxygen revolution, which means you have non-cyclic photophosphorylation evolving, water is getting split, and oxygen is getting released. Okay, you're still in the Precambrian period. 2, 3, 4, you're still 4 in the afternoon, you're in the Precambrian period, and now eukaryotic cells have evolved from prokaryotic cells, probably due to the endosymbiotic hypothesis. It's 5, it's now dinner, 6, 7, 8. It's not till between 8 and 9 at night that you had your first multicellular organism, and now we're finally shifting into the old and Paleozoic era, right? So formation of the earth, this is 4.6 billion years ago. Go, 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 go. And now dinner time, right, is when the Paleozoic era starts. If you look at your notes on your Paleozoic era, right, that was about 542 million years ago. So this is when you get your land plants. And then look, Mesozoic era, it's like 10:30 at night, right? That's about 248 million years ago. Cenozoic era finally starts, okay, about 65 million years ago. And then look at the time. It's 30 seconds before midnight. So we've gone our whole 24-hour clock minus 30 seconds. That's when you start to see man and those types of fossils, Homo sapiens, on the planet. So hopefully that gives you some perspective where 1 second equals 52,000 years. All right, now let's break down the tree of life. So here's your first cells, right, about 3.5 billion years ago, and you can see the time scale along the bottom here as well. Remember, we have our 3 domains, bacteria, archaea, and eukarya. So here we have bacteria, and here we have archaea, and you have, from archaea, you have these different categories of archaea that we've probably talked about, but I want you to look right here. Branching off from archaea is eukarya. Eukaryote is you and I are built out of eukaryotic cells. You can see a little bit of the endosymbiotic hypothesis where some bacteria went to live into other bacteria and formed heterotrophs or autotrophs. Okay, now this is still, look, 2.2, 1.4. Now, right about here, we're starting to transition into the Paleozoic era from here. All of this right here, all of this would be during Precambrian times. So Precambrian times listed by billions of years, formation of the earth, oldest known fossils, oxygen accumulates, you get eukaryotes, protists evolve, and then you get these Ediacaran fossils, really flat, soft-bodied invertebrates. I will show you a picture of them momentarily. Look right here. I just want to remind you again about the endosymbiotic hypothesis. We've talked about that before. And then here are these Ediacaran, I hope I'm saying it, Ediacaran fossils. These are they're multicellular and very thin and very flat bodied. So in Precambrian, what I want you to put on your notes is you went from the first cells all the way to invertebrates and algae. From the first cells all the way to invertebrates and algae. Okay, so take a look. That was Precambrian, but that is the bulk of the time, right? Remember Paleozoic, we were like 8, 9, 10 o'clock at night when we started this, right? So we're going to go through, okay, the rest of this now. So let's talk about the Paleozoic era. A lot happened in the Paleozoic era, which obviously this artist caught, okay? You went from algae all the way to gymnosperms. These are things like with pine trees, pine cones, and you went from these soft bodied worms all the way to reptiles. So on your notes for Paleozoic, I want you to go from algae to gymnosperms, and then you went from invertebrates all the way up to early reptiles, invertebrates to early reptiles. All right, so here, you started to get a lot of insects at this time. This is great. This is going to be great for future for pollination later. That we're going to get to that. But those are starting to evolve at this time. Now let's get into the Mesozoic era in the middle, right? This is between if you go back, remember, look on your notes, Mesozoic, you're at 248 is when this starts, and it ends about 65 million years ago. You're in the middle. And in the Mesozoic era, you're probably familiar with all the dinosaurs that evolved at that time. So as far as the animals go for the Mesozoic, you want to go from these early reptiles, and we are going to go from these early reptiles all the way up eventually to mammals. I'll get to that picture eventually. Early reptiles to mammals. And as far as plants go, you go from gymnosperms, like those Christmas tree kind of pine cone or Hanukkah bushes. You go from gymnosperms all the way up to angiosperms, which are flowering plants. Okay, gymnosperms all the way up to angiosperms. Okay, and then here's your early mammal. Maybe you remember a movie where it looked like this. Okay, the Cenozoic era, the era that we are in now. And yes, that included the woolly mammoths. So from there, you're going, you can recognize maybe some of these a little bit. You're going from those early mammals and you're working your way all the way up to humans, early mammals to humans. And then as far as plants go, you're going from those early angiosperms all the way up to herbs, just Herbs. And we'll just leave it at that. All right. So I just want you to see on that biogeographical, you just need to know like the bookends and then you should be fine on that. For instance, I want to go back to when we talked about, go to your Paleozoic era when I talked to you about going from invertebrates all the way to reptiles. To get from an invertebrate to a reptile, invertebrates, you have to have vertebrates, and then you would have fish, and then you would have amphibians, and then you would have reptiles. So I just told you the bookends, but you've got to evolve through all of those creatures to get there, all right? So next, one piece of evidence is fossils. So we've gone over fossils and we have looked at the geographic timescale and looked at how those fossils have changed over time. Our next piece of evidence is biogeographical evidence. And so this is the idea. Let's go ahead and put it in your notes first. It provides evidence that variability in a single ancestral population can lead to adaptation to different environments through the forces of natural selection. It is driven by competition. Okay, can lead to adaptation to different environments through forces of natural selection. It is driven by competition. So you're going to adapt to whatever environment you're in. And if these plates are moving around and changing, just like the horse was initially adapted to a forest environment, but as the plates move, then it became adapted to a grassland environment. So whoever's going to survive in that environment is going to be the one that survives and reproduces and makes more species. So a really good example of this is mammals. Remember, early mammals were starting to evolve about 65 million years ago. And this was at the point where, remember, the continents were all together, supercontinents, Pangea. But by about 65 million years ago, all the continents were separated from each other. So because they were separated from each other, when mammals evolved, they're very unique to each of our continents, right? We don't have giraffes running through North America unless they were released from a zoo because they didn't evolve there, right? And so the oceans provided a barrier. And at that point, you're not having any shared evolution. Now, when the continents were all together and let's say ferns were first evolving in the Paleozoic era, then you're going to see it be continuous through groups and multiple continents because all those continents were together and so it could spread around. So on your examples for biogeographical evidence, when continents were joined, there were similar species, but after separated, more continent-specific species like, say, marsupials in Australia. Okay, so that's another piece of evidence. The continents were together, kind of have that uniform evolution, but when they're separated, you see very, very specific to their location. All right, next one we want to talk about is anatomical evidence. And with anatomical, you're looking at anatomy, and there's 3 bits of anatomy that we're going to look at. The first one we're going to talk about is homologous structures. Homo means same, like homologous chromosomes, homologous structures. So if you look at your arm, you have your humerus right here. You have your radius and your ulna. So your radius, you'll always know is, sorry, I'm not showing you very well. Your radius is connected like directly to your thumb, and you can see how it moves over your ulna. And then you have your carpals, metacarpals, and phalanges. So you can see that same bone structure in a cat. Humerus, radius ulna, carpals, metacarpals, and phalanges. You can see it in a whale because they are also a mammal. Humerus, radius ulna, carpals, metacarpals, and phalanges. You can see it in a bat. You can see it in a bird, right? Just like a human. So this anatomy is the same, but yet we do very different things with these structures, right? Some are used for flying, some are used for swimming, some are used for walking, some are used for grabbing. But what does it tell us? If we have the same anatomy, then we probably have the same DNA to code for that similar bone structure, which means we probably have a common ancestor. Now, do not be fooled by analogous structures. Analogous structures, and here I will show you, analogous structures are similar in function. This is not evidence of evolution. It just means you're in the same environment, and so you have to deal with moving through that environment or living in that environment in the same way. For instance, bats and butterflies both have wings, but the structure of a butterfly wing and the structure of a bat wing or a bird wing are very, very different. So they are analogous. So you can't look at that for evidence of evolution. It's just the environment that they are in and what they are adapting to, right? So for instance, this bat and bird, they have the same anatomy. They are homologous. They just so happen to be analogous as well. A bat is not analogous with a whale, okay? But yet it has the same anatomy, so that points to a common ancestor. So on your anatomical evidence, homologous structures are similar anatomy, but may have different functions. And I said, think vertebrate forelimbs. And then I gave you analogous is similar in function, and it is not evidence, okay? Vestigial structures. These point to our ancestral history, where we came from. For instance, whales all have part a pelvis and part of a femur. Well, what do you use a pelvis and a femur for? Walking. So that tells you in a whale's ancestral history, right? He must have been walking on four legs, but moved into the water for whatever reason, better survival. And so you still have remnants of this femur and this pelvis, but because it's not important, it's not selected for because it doesn't help him survive. So you just see little bits of it. Let me give you an example of this. Here's a salamander. He has vestigial withered eyes. So what does that tell you? So I'm putting a dot right here, okay? So at one point he had eyes there. Why? Because he lived out where he needed to see his environment. But let's say he started living next to a cave, right? And then a little bit farther in the cave and a little bit farther in the cave, he didn't need his eyes anymore. So that was not hinging upon his survival. So if he had a bad eye, he was still having sex because you didn't need a good eye to get a mate, because it was dark in the cave. So that points to your ancestral history, but it also gives you evidence that things do change over time. All right, so on vestigial structures, anatomy traces evolutionary history. Anatomy traces evolutionary history. Think vestigial eyes in cave salamanders and whale hind limbs, or probably our appendix. It probably helped with digestion in our ancestral past. Okay, then the third piece of evidence relating to anatomy is embryological development. Couple of things here. One is similarities in embryos would tell you probably more closely related that they are, but also when embryos develop, it's like evolution on fast forward. At one point, we have a tail. Okay, we have a tail. At one point, we have gills in our development as an embryo. So this is kind of like we're going through all of our evolution really quickly in our womb. So embryological/developmental structures, all vertebrates have a postanal tail and paired pharyngeal pouches, which become gills in fish. So that is the third piece of anatomical evidence, okay? So we've had fossil evidence, and we looked at the geologic timescale. We've had biogeographical evidence, and we've had anatomical evidence. The next one I want us to do is biochemical evidence. And this is where you compare, in this case, we're comparing the number of amino acid differences in cytochrome C, okay? Now, Do you remember mighty mitochondria and cellular respiration and along the electron transport chain, right? Reductase, cytochrome oxidase. Do you remember that? So cytochrome C is a small protein. It plays an important role in the electron transport chain within the mighty mitochondria. So how similar is that protein? Well, not a lot of difference between us and a monkey, a little more difference between us and a pig, a little more between a duck, a turtle, a fish, a moth, and yeast. So when you look at this, the assumption you make is the more similar our proteins are, the more closely we are related, meaning we probably diverged from the pig line more recently than we did from the fish line, right? And that makes sense because a pig is a mammal. So biochemical evidence, almost all living organisms use ATP, DNA, and specific enzymes. ATP, DNA, and specific enzymes. The more similar the DNA/proteins, the more closely related. So you can use it like a molecular clock. It uses comparisons of DNA sequences to estimate phylogeny and rate of evolution, meaning how fast are you doing it. The differences of the genes indicates the presence of mutations. The more mutational differences, the more time has passed. The more mutational differences, the more time has passed. And the rate of mutation can be affected by type, location in the genome, and the populations affected. So you can compare proteins, you can compare DNA. This is an example of, look, human, monkey, pig, cow, rat, mouse, right? And these are where you see differences, okay? So this would be a mutation, a difference in the amino acid that's building this protein. So if the amino acid is different, that means you have a mutation in the DNA that caused that difference. All right, so the greater number of shared DNA sequences between species, the greater number of shared genes, and therefore the greater the evidence that there is a common ancestor. So when you talk about that molecular clock that I just mentioned and gave you the notes on, you're comparing DNA sequences, and you're doing this to estimate its phylogeny, who you could have evolved from. So the difference between the genes indicates the presence of mutations. The more mutations they have, the more time has passed. And the rate of mutation is affected by many different factors: where it is in the genome, the type of mutation that it affects, and the population. So look here, this could be your ancestral genes. And then over time, okay, you have some mutations. So this guy mutated, you've got a red and a purple, and this guy's got a gray and a blue. And then a little bit more time and a few more mutations. So this is species 1. These two, you can tell that they are related. Why? They have the same mutation of that color, is just representing the DNA, right? That same mutation of gray and blue, but they have two different mutations here. So you can look and compare the number of mutations to build back your evolutionary tree where you came from, and that's your phylogeny. So the classification of phylogenetic tree for a group of organisms, you want them to construct who are you related to. So for instance, a bear is more closely related to a giant panda. Red pandas are more actually related to raccoons than they are giant pandas. So just because somebody names something some way doesn't mean that they're actually related to one another. So systematics is the study of the diversity of organisms using information from the cellular level, cells, all the way up to populations, and how they interact with one another. And so phylogeny is tracing the evolutionary history of an organism. You're developing a phylogenetic tree. So underneath phylogenetics, it's a modern way in which organisms are classified and arranged in evolutionary trees. When a new character or trait evolves, then the tree branches. That is at a node, and this is visualized by cladograms. I know this is kind of a long video. I didn't mean it to be so long, but let's finish it up. This is our last topic, is cladograms. So cladistics, what cladistics does is it tries to arrange based on evolution. How do you know? You're comparing DNA, you're comparing proteins, you're also looking at structures, any kind of character, to arrange them based on their evolutionary tree. It's kind of like being in a club. So for instance, a hagfish is a jawless fish. okay? Every organism, from the perch all the way to the chimp, they all have jaws from here on up, okay? But now everybody except the perch has lungs. So everybody is in the lung club from here on up. These animals are more closely related to each other because they have this characteristic, and so forth. So how do you do that? Well, you have to look at characters. A character is any structural, chromosomal, or molecular feature that distinguishes one group from another. So it could be anatomical, but it could also be your biochemistry within your cells. So a cladogram, what it's going to do is it's going to reflect your evolutionary history. So put that in your notes. It reflects the evolutionary history / relationships. Okay, it is the common ancestor, and all of its descendant lineages are called a clad. And I'll show you how you go about doing that. All right. So I think we did this. So you look at two kinds of characteristics. You look at what is the ancestral trait that we all have, and then what are the derived traits that not everybody has. So for instance, all of these organisms here, they all have vertebrae. So that's their ancestral trait. And now we're looking at anything that differentiates them. So from here on up, they have a bony skeleton. From here on up, they have 4 limbs. A node is where a branching occurs, and like you're no longer in the club and everybody else is continuing on. So your ancestral characters are those that are found within the entire line of descent of a group of organisms. And then derived characters, that would be all the rest of these characters. Those are present in members of one group of the line, but not in the common ancestor. So let me show you how you would do that. Now, I've set this up so it'll be easy, okay? But you would look at a bunch of, you would have all of your organisms here, and you would list off all the different traits you see in all of them. You'd list them all here. Not all of them may have every single trait, right? But you just list all these different characteristics. And you look at for something that they do all have, the ancestral trait. In this case, all of these organisms have a backbone. Okay, so that will be our ancestral trait. Check, you got a backbone. And then we're going to build our clad from there. So how many of these organisms have lungs? All of them, right? Except the fish. So we would put checks here. Okay, so we're going to be dumping out the fish here, but from the salamander up, we get to continue on in our cladogram. So you would go ahead and fill this out. I've arranged it so it's really obvious. I know that. Okay, and then from that, you could build your cladogram. So here's the beginning of my cladogram, and here's our little checkbox that we used. So the ancestral trait is the backbone, right? Then what is it that not everybody else had? Most everybody had lungs, right? Just not the fish. So our next trait we're going to put right here is lungs, because everybody else in our clad has it, and we're going to put the fish right here. So I'll show you that. Okay, so here's our backbone, our ancestral trait. Then our derived trait is lungs. And fish, he doesn't get to keep going with us. And we would continue on placing these. The next one, right, would be claws, because everybody has claws except our salamander right here. And then everybody has hair except the lizard. So you would eventually build your cladogram like this. Now, this is a very simple kindergarten version of a cladogram. They obviously get more complex when you're comparing DNA sequences and amino acid sequences in order to develop your cladogram, to look at slight differences between, let's say, primates. Or if you're looking here, you're using biochemical evidence to construct this cladogram. And so what are you looking at when you're, what are you looking at is you're looking at, you're comparing DNA in order to, instead of saying, oh, you have hair or, you have nails, you're actually looking at the sequence of the DNA in order to, or the proteins within them, in order to organize your species and discover the phylogeny. Okay, long video. That is the end of part 1. Part 2, we will do in a second video, and I'm stuck right here, on the process of evolution. And I hope you're having a good day, and that's it.